So, a very good morning to all the participants. Today, I'm going to present my topic, that is microbiogeochemical factors responsible for arsenic mobilization and isolation of heavy metal hypertolerant bacterium from irrigation well water. Well, I am Dr. Abhishek Basu, and assistant professor and principal investigator. My affiliation is Department of Molecular Biology and Biotechnology, Sipat Singh College, affiliated to University of Kalyani. For this work, my co-authors are Devashish Chatterjee and my PhD student Devjani Mondol. I'm glad that I have been given an opportunity to present in 12th ICSTR Singapore International Conference. Yeah. So basically, our area of work. This is our area of work. This is Chagda block of Nodia district. This is a district of West Bengal, India, which is situated in Bengal Delta Plain. As we know that Bengal Delta Plain has a problem of arsenic contamination of soil and groundwater because of the presence of arsenic in the aquifers, and its origin is completely geogenic. So, groundwater is commonly used for drinking and irrigation purposes throughout Southeast Asia, notably in Bengal Delta Plain, that is mainly in West Bengal and parts of Bangladesh. In major part of Bengal Delta Plain, shallow aquifer, which is less than 50 meters groundwater, is used for irrigation purpose during paddy cultivation, because paddy is the staple food over here. Also, the summer paddy, which is known as boro, has been produced with large-scale groundwater development up to 90%. Our present study focuses on arsenic mobilization in shallow irrigation well with analysis of water quality as well as related geochemistry of multi-depth wells, that is from 15 to 42 meters. Okay, and these are all irrigation wells. Here are some red spots. As you can see, these are the location and distribution of the irrigation wells which we have sampled. We have collected water from them and did a details biochemical and microbiological analysis of the same. So, due to the time constraints, I will only show to you the main observations. Here we could see that there is a strong correlation between arsenic and iron concentration. Okay, so which suggests that iron oxide hydroxides could be the possible host for the release of arsenic. Okay, they actually host arsenic, and this dissolution of iron minerals release arsenic in the groundwater. And the anoxic as well as reducing condition of the aquifer supports this kind of release of arsenic from various iron oxides and hydroxides minerals which also bears arsenic. Also, this is very interesting what we have seen, that in the irrigation wells there is presence of coliforms and fecal coliforms. Okay, now this irrigation well water is mainly used for irrigation purposes, sometimes for consumption and drinking or water. Okay, if they are fed to cattle. Okay, but if they contain coliform and fecal coliform bacteria, which is very harmful for health and may cause fatal gastrointestinal diseases, it's a matter of concern. To further confirm the presence of this bacteria, we did fermentation test by culturing whatever microbes present in the irrigation well water in lactose broth, and we could see the gas bubbles here over here in the Durham's tube. Also, there are presence of fecal coliforms, and these are specifically belongs to the class E. coli with this green colony with metallic sheen, and here are the pink colonies of Enterobacter aerogens. Okay, so here we can see First, I'll explain for the estimation of total coliform. We cultured uh, the irrigation well water in lactose broth at 37 degrees for 24 hours. But for to see whether fecal coliforms are present or not, we cultured the same irrigation well water at 44.5 degree Celsius for 24 hours. We could see here that there are various wells, okay, which actually contains both fecal coliform as well as normal coliform bacteria. However, this well, it contains only coliform and not fecal coliform. But you could see well number 15, 16, and 32, 36, and 40, they contain both coliform as well as specifically fecal coliform. Okay, in the well number 39, we could see total coliform but no fecal coliform. And also, there is a correlation between the presence of coliform and chloride ion concentration. Okay, so here the horizontal axis shows the number of irrigation well, and the vertical axis shows the concentration of total coliform, fecal coliform, and chloride ion. We could see wherever the chloride ion is having a higher concentration, there is presence of this coliform bacteria. Uh, Specifically, obviously, total coliform and some fecal coliform. Now, this chloride ion could have anthropogenic sources. This is very important because what we have seen over there is leakage from sanitation feet and sewage line, which may contaminate the groundwater. As it is contaminating the groundwater, maybe the fecal coliforms or the other coliforms present in the sewage water, okay, they are actually getting transferred to the irrigation well water and contaminating the same. That's why there is a positive correlation between the concentration of chloride ion and the presence of coliform bacteria. Now we have isolated this bacteria from one of the irrigation wells of Parari village. So this bacteria is highly arsenic tolerant. Okay, in increasing concentration of arsenic up to when we have used 100 ppm of sodium arsenite, it could grow very easily. Even in 2000 ppm of sodium, in 1000 ppm, 1500 ppm, 2000 ppm of sodium arsenite also, this bacteria could grow very easily. And this is the electron micrograph image, electron microscope, not micrograph, sorry, electron microscope image of this bacteria. So it is almost 1.2 micrometer in size, and we have called it DACW based on the initials of our own name, and it gives round shaped white mucoid colonies in this kind of LB agar plates. Further, we have characterized this bacteria using 16S rDNA sequencing followed by detailed phylogenetic analysis. So this phylogenetic tree has been constructed by using the neighbor joining method, and the evolutionary distances were determined using Jukes contour method. What we could see, this DACW, the identity of this DACW is Enterobacter sp because it's the closest homolog strain NA11039. Okay, so finally we have isolated arsenic hypertolerant bacteria, which shows extreme tolerance to both arsenite as well as arsenate. And this is the growth curve of this bacteria. Um, you could see that in 100 ppm, 500 and 1000 ppm of arsenic, it is hardly showing any kind of inhibition of growth. Okay, but in 2000 ppm, which is really a very high concentration, and that's almost nearing its minimum inhibitory concentration value as well. Okay, at that concentration, there is a slowdown of growth, but still it is growing at that high concentration also. This is in presence of arsenite. Now, arsenite is the most toxic form of arsenic because of its high solubility, mobility, and bioavailability. However, arsenate is comparatively 100 times less toxic. Also, this bacteria, since it's growing, this bacterium, since it's growing at such high concentration of arsenic, there could be a chance that it is either um, using arsenic for its metabolic purposes and which is releasing energy. It's using arsenic to generate energy. So, which is kind of a dissimilatory process, or it is assimilating arsenic within its cell, or there could be an interplay of both this dissimilatory and assimilatory processes. What we could see next is that uh, this bacteria could biotransform arsenite into arsenate, the much less toxic form. Huh. And this is one step of bioremediation of arsenic. And this is actually a figure is shown in the LB agar plate where we have cultured this bacteria and put in presence of sodium arsenite, obviously, and we have put silver nitrate. So this is the like turning brown. This is the colony which is now brown in color. It turned brown because of the formation of your silver arsenate. Okay, since it is converting arsenite to arsenate, okay, now this arsenate is reacting with the silver nitrate and forming this brown color columns. Now, this ability of biotransformation could be further used for bioremediation of arsenic from groundwater, soil, or wastewater. So the results they suggest that the groundwater is usually anoxic with high amount of dissolved redox elements, specifically arsenic and iron, where the iron concentration is much higher compared to the arsenic concentration. Now the second point of our result, which I haven't shown much in the data format, it's the high concentration of alkaline metals like calcium and magnesium along with high alkalinity suggests the carbonate dissolution is the most important mineral dissolution process in the shallow aquifer. Again, as we could see in the third result, the arsenic and iron, they have a positive correlation, and also high chloride ion concentration in some of the wells indicates towards local anthropogenic influence like leakage of sewage. The study also reveals the presence of microbial community like fecal coliform in the monitored irrigation wells with high chloride ion concentration. The presence of fecal coliform further confirms the role of anthropogenic activities like sanitation, open dumping, organic carbon source, and faulty construction of wells. Uh, some irrigation wells with non-permissible concentration of arsenic, yeah, that's beyond the which is the maximum permissible concentration by BIS and WHO, that is 10 ppb. Okay, some of the wells contain much more than that, and from there we have isolated this arsenic hypertolerant bacteria, and we have characterized the same using 16S rDNA sequencing and phylogenetic analysis and scanning electron microscopy. And this turned out to be Enterobacter sp, which could basically biotransform arsenite into arsenate. So the basic conclusion of this study is that the influence of anthropogenic activity on natural processes, which increase the reducing environment of the shallow aquifer and also release arsenic in the groundwater. Now, this work is funded by two projects sanctioned by Department of Science and Technology and Biotechnology, Government of West Bengal. Both the projects are actually directed towards the bioremediation of arsenic from the arsenic infected soils of Nadia and Murshidabad district, as well as to provide a safe source of groundwater, which is also used for drinking purposes in those regions. I thank the organizers for giving me this opportunity to present my work. And if there is any question, I'll be happy to answer.